Let's start. Okay, let's start. Welcome to the last class of this course. And it's our 10th meeting. And today we will speak about interaction between growth factors and monoamines. It's a very difficult topic. It's actually this topic deserves full course, but we will try. We will speak about three trophic factors. One, we briefly will speak about BDNF, but BDNF you can speak, again, you can speak full course about BDNF. Then we will speak about fibroblast growth factor 2, FGF2. It's, and then we will speak briefly about, and then we will speak about transforming growth factor beta 1. And let's start with BDNF. I'm sure that you all heard about BDNF. BDNF is a neurotrophin, is a neurotrophic factor. It's a relatively small protein of 14 kilodalton. It's fundamental, it's involved in all aspects about neuroplasticity. And also neuroplasticity is a very complex topic. There is no simple answer what is a neuroplasticity. Neuroplasticity is its ability of the central nervous system to adapt itself to the for the purpose for the more appropriate response to the environmental stimuli. And if you speak with neurohistologists, neuroanatomists, they will see neuroplasticity in the term of neurogenesis, growing of dendritic trees, or creation or remodeling of the dendritic spines. If you speak about molecular neuroscientists, they will speak BDNF and other nerve factors involved in neurons. If you speak about electrophysiology, he will speak mostly in the terms of the activity-dependent synaptic plasticity. But in all these aspects, literally, and also in electrophysiological, in neurochemical, in neurohistological aspects of synaptic plasticity, BDNF is involved in all. But BDNF is not what you may not, some of you may not hear. BDNF is not only brain trophic factor. BDNF has also important function in other organs, and specifically BDNF is pretty important in the female reproductive system. It's involved first in folliculation, when oocyte leaves maturate and leaves the ovary, and it's also involved in the implantation of the embryo and development of the placenta. And about, as I told, we need BDNF for neuroplasticity, and we need neuroplasticity for the treatment of depression. And literally all treatments of depression you can summarize by this. It's, for example, pharmacotherapy or physiotherapy like electroconvulsive therapy, or even physical exercise. We saw in one of the previous classes that physical exercise, voluntary physical exercise, has a good pretty potent antidepressant effects. The mechanism of action, or more or less, can be simplified as follows. First, you have increase in monoaminergic neurotransmission, and this increase in transmission happens because of desensitization of presynaptic receptors or upregulation, sensitization of postsynaptic receptors. When you get increase in neurotransmission, you get increase in expression of BDNF, and then you have antidepressant effect. We also learned before that BDNF knows to reinforce its own expression. In that two loops, there are short loop and long loop of reinforcement. Short loop BDNF stimulate expression of VEGF, VGF, it's a precursor of several peptides. TLQP62 is a C terminal of this VGF, which get cut and act as act also as a neurotropic factor, which stimulates back release of synthesis of BDNF. And long loop involves monoamines, mostly serotonin. Serotonergic input stimulates expression of BDNF, and it's possible. Actually, I get involved in BDNF recently, and I don't go to do work in which laboratories and very important scientists did a lot of work about effect of BDNF on neurogenesis, neuroplasticity. I would like to what I would like to check what we are working about, if there is positive feedback. If BDNF, after it's stimulated, after it's triggered by serotonin, if it's enhanced back serotonergic transmission. There are some evidences for this, and we are working now on this hypothesis to check if there is this long loop. BDNF stimulates excitability probably of serotonergic neurons, which act back on expression of BDNF. We also, it's one of our topics, what we are working out, how BDNF signaling is connected with calcium signaling, with NMDA receptors, and with voltage dependent calcium channels. And what was our focuses? I think when we have virtual visit to our institute, you saw Dr. Gabriakos, and they were the unique method of research of ryanodine receptors. I think we also show this figure before, that how antidepressant works. You start to take antidepressants, you get increase in the serotonin, you get activation of autoreceptors, inhibition of serotonergic neuronopharyngeal activity. Then within 2 weeks, you have sensitization, adaptive changes, 5-HT1A and 5-HT1B receptors desensitize. You get increase in serotonergic activity, which leads to increased BDNF expression. And also we are asking question if it's all work backup. We think that they are, and through creation of this positive reinforcement loop is very important for to see the clinical and behavioral effect of the treatment. And we also ask also very kind of pretty, how it's called, if you can just bypass it straight away to administer BDNF or TLQP. And indeed it was done in one research group did it. Yes. TLQP is this self-reinforcement of BDNF synthesis. BDNF, it's C-terminal off of VGF, which enhance synthesis of BDNF back. Okay. They implanted cannula to the prefrontal cortex of animals. They allow to animals to mice, I think, to recover 7 days. And they administered the TLQP straight to the brain, because it's a peptide which is not passing blood-brain barrier. And they get very strong antidepressant-like effect in open field and in forced swim test. But there is a small problem with this, and because not every depressed patient will allow to drill to himself the head and to implant cannula, and he will be right not allow it. So how we bypass? What can we do? How we can deliver to the brain large biological molecules like peptides or like even better nucleic acids. And the answer is one of the ideas. It's nasal, to use nasal route. And in case if you use peptides, you can just administer them nasally. You will see by end of today's class our first successful experiment with the nasal administration. And but if you administer nucleic acid, like to deliver to the brain BDNF mRNA, then you need to, of course, to protect it because nasal cavity, like even more than skin, is full of RNases, and it will not be successfully delivered if it's not encapsulated and covered. But development with nanotechnologies did it possible. And we are collaborating with the laboratory, with the Polymer Institute of Slovak Academy of Sciences. It's one of the best institutes of our academy. And there is a laboratory which specializes in polyplex in these polymers. They create tissue-neutral polymers, which does not make any interaction with the immune system. This is cationic, positively charged polymer, and because mRNA is negatively charged, it creates capsula protecting. And this capsule, these nanospheres, we are trying to administer via nasal cavity. Next step will be to attach to this polyplex nanoparticles antibodies, which will connect them to the neuronal transport proteins, to deliver them using this method from the nasal cavity to the internal structure of the brain. What is very good, that brain is that olfactory system very densely interconnected with limbic areas of the brain and with monoaminergic nuclei. Excuse me? Yes, it's small, yes, and relatively to if you cough, all of you saw rodent brain, rat or mice brain. You can, of course, of olfactory bulb relatively to the huge relatively to the brain, and our olfactory bulbs just a little bit bigger than in rodents, but in comparison to such a big. But of olfactory system evolutionary oldest one, and it's very similar. I think it's almost identical in rodents and in primates. But what is good with there is one antidepressant which approved for nasal administration, esketamine. Now we finished a pretty time-consuming. You see, we tested several polyplex. And first of all, we created a polyplex with these polymers and mRNA synthesizing for fluorescent GBG protein, fluorescent green protein. And we checked finally we choose the best molecules which are good in terms of penetration to the neurons and which are less problematic. And for some polyplex we did not proceed with it because we saw the toxic effect. With some we did not see appropriate penetration, but what's for the last one is till now the best one. You see you see very beautiful neurons. It's the neurons in primary culture, and you see the penetration of this polyplex and expression of fluorescent green protein. And sorry, here you see pretty toxic effect.
No, but the third one seems just fine.
We are working with the two last ones. Yes. Now we did first in vivo experiment. We took this polyplex and applied it nasally to the rat, but I don't have yet results. We're about soon to cut the brains and to see it. Let's quickly about, I told you in the beginning that BDNF is not only about the brain, that BDNF is also important in peripheria, and we will try even to connect to understand how peripheral BDNF, which are working on the which is very important for that in a female reproductive system, how it can be connected with that central BDNF. I will upload this. This is a review article which I also took from it. I will later upload it. BDNF, maybe you already read similar articles. BDNF can be detected in plasma outside. It's not very clear where it's coming from. It's more or less clear. There are studies that central BDNF leak out to the periphery, and peripheral BDNF also can arrive to the brain back, but less. Why less? Because we have drainage of our CSF to the circulation. Together with the drained CSF, we also get peripheral BDNF. But BDNF also synthesized in the periphery. BDNF can be synthesized by platelets, by BNT cells, and by vascular endothelium. What's happened with the peripheral BDNF during pregnancy? During pregnancy, you have always decrease in humans and in rodents. You have decrease in pretty drastic decrease in BDNF, which recover after the birth. If you have too much decrease in BDNF and something happens, some failure happens here, and there is no proper recoverable BDNF, it's probably one of the reasons for postpartum depression. And why we have probably increase in cortisol during the pregnancy, we have increase in cortisol, which leads to peak by end of the pregnancy. Where maybe it's one of the factors, because cortisol is a stress hormone, and pregnancy is not illness, of course, it's normal physiological situation, but it is stress. Then we have it's natural to have decreases in BDNF, and also increase in estradiol, probably is also one of the factors which suppresses BDNF. And as I told, BDNF important not only in the brain, but also it's also acting in the ovary, in the uterus. This is just peripheral BDNF. There are plenty of studies showing that it's in very strong correlation with postpartum depression, which means that it's not just leak out, which means that peripheral BDNF concentration, it is informative. And it is important in brain function. Maybe it's not important itself in the brain function, but it suggests about central levels. And
or is it you?
what? It's a review.
It's not your.
No, it's a review about many.
I suppose the BDNF can be lower. Can be low in some certain people, right? Like
It has a ratio different between it was they found, for example, different between races. In African American women, they have more vulnerable to decrease in BDNF than Caucasian. Yeah.
So my question is, does it matter how does the BDNF need to go back to normal to not to?
I think so.
Or just generally high? Like, I don't know.
It's okay.
It's like
It's okay. It's normally during the pregnancy. It's normal that BDNF is going down. It's happening in every woman.
So from what I understood, you said that those who had postpartum depression, their BDNF stayed down.
Yes, it's going down more than usual, or it stay longer down.
Okay, so my question is does it have to be in a certain, does it need to be completely normal? Does it need to like spike up for it to not, I don't know.
Well, we don't really know. I think pregnancy is also, as I told, is also stress. I think when you have pregnancy plus another stress before pre-gestational or prenatal, and then you probably have too much decrease in BDNF, and then you have postpartum, then you may have postpartum depression or even prenatal. There are much less studies about prenatal depression, but it's also a common condition, especially in the last trimester. Probably it's also connected with decrease in BDNF. And what is and actually postpartum depression we speak when woman after the birth severely depressed. But at least sounds like depressive symptoms I experienced nearly almost by every woman after the birth. Probably this crisis that shut down BDNF, this is what's and as I told, there are studies you can also found in these reviews. Yes, please. There is one study says that yes. We will soon come to the one slide, which I think will give to your answer to your question. There are studies also observation that low maternal BDNF and maternal BDNF go too much down. It's increased risk of preterm birth. It's one of the risk factor for low of normal, like when pregnancy, length of the pregnancy is normal, but weight of the baby is too low. And also it is known that it decreased if peripheral BDNF level are low, it's decreased the success rates of IVF. It's why it's well known by doctors who is caring with IVF that stress is one when a woman's too much stressed. It's the time of IVF. It's really decrease the rate of the.
Regular successful pregnancy is like, okay, we'll take that low BDNF.
I think so. Yes, you see in these studies they found correlation between the scores, between the severity of symptoms, and between levels of circulating BDNF.
Yeah, but if depression in a low BDNF, probably to some problems in the reproductive system is with anyone with depression and lower successful pregnancy rates. It's taking it one step further.
I don't know, really. It's a very interesting question. We can make search and yeah. We spoke about already in the previous classes that maternal stress and maternal depression affect development of the brain and reprogram the activity of 5-HT neuron, for example, of 5-HT dopamine neurons. What we saw in the last class, that when you have imitation of the sickness in pregnant rats, gestating rats, offspring characterized by decreased activity of serotonergic neurons, and these offspring, from other studies, they are more anxious, they're more vulnerable to anxiety. You will be very surprised that if you take another types of stressor and apply it on different time on the mothers, you have totally different results. For example, this is another study from our lab, and we checked in collaboration with Institute of Pharmacology, and we checked prenatal pre-gestational stress and prenatal antidepressants on the serotonergic system in offspring. And what you see is that in male offspring, we saw with a previous stage, with the other type of stress or another time of administration, not during the pregnancy, but before pregnancy, you have even increase in serotonergic firing activity, which is probably kind of adaptive mechanism. And you have summary, for example, from studies from different laboratories, maternal immune activation is we saw decreased serotonergic neuronal firing activity and decreased glutamatergic activity glutamatergic neurons in hippocampus. Chronic unpredictable stress when you apply progesterone will have other effect. Restrained stress when you apply during the pregnancy, it's very similar to our favorite 5-HT system. And the question, I personally very curious what's going on, what's mediate, what is actually pathway which connected maternal brain with embryonal brain. And our hypothesis that it might be BDNF. We learned that BDNF is leaking out from the brain to periphery, and peripheral BDNF levels, they are corresponding to central BDNF level. Probably when mother is stressed and central BDNF goes down, probably also peripheral BDNF is down. We know that placenta is very rich by tyrosine kinase B receptors. BDNF is unlikely to cross placenta. But what it can do, it can stimulate VGF synthesis in placental cells via these receptors. But BDNF circulated in maternal blood maybe activate tyrosine kinase B receptors, and maybe it triggers increase in embryonal BDNF and triggers embryonal. And again, the side of the story was topic of grant application, which I also get informed that it was not approved. So we go to resubmit it to another call. Right. Some question was to BDNF. Okay, let's speak about fibroblast growth factor 2. I think Hila can speak better than me, maybe. You don't have told Hila today? Okay, that's no matter. Fibroblast growth factor 2, again, it's a protein. It's a primary function. It's proliferation and activation of fibroblasts. But it's also involved in plenty of the central nervous system processes. When we speak about primary function, actually, we don't know what is its primary function, because first it was discovered in fibroblasts, and why it gets its name, but it's hard to say what is in nature. It's hard to say what is actually main function, what is and what is side one. What was found by this is what was discovered that FGF2 has a pretty potent antidepressant-like effect. It's correct stress-induced anhedonia, and it's correct stress-induced neurogenesis, adult hippocampal neurogenesis. But I actually get introduced to FGF2 by Professor Segev Barak, faculty member of this school. And I heard before I even knew that the FGF2 can be a good guy, can be potential antidepressant. In Tel Aviv, we work on the FGF2 from the other aspect. Unfortunately, because my rules have negative effect, it's involved in alcohol consumption. It what is increase and stimulate alcohol consumption. And what is the neurotransmitter basis of alcohol use disorder and all other, and all very simplified very quickly, because it's also very deep topic about neurotransmitter basis of addiction. But basically there are two main components. First is the reinforcement. It's willing to re-experience the high, the pleasure, is the hedonia created by drug or by alcohol. And other component is the impulsivity. It's the ability to stop yourself and to restrain, to not to follow immediately your pulse for you reward seeking. Probably FGF2 works on this, should be connected to these two systems. And we started to work with Segev Barak, started to work on FGF2 and dopamine system. And it got published a year ago. We indeed found that FGF2 increases the firing activity of dopamine neurons in the VTA and also in substantia nigra, which I am not showing here. And we also checked which receptor is mediating it. FGFR1, it's the FGF2 receptor 1, is one of the receptors which modulates FGF signaling. And we found some that this was done on the left side, it was done in Tel Aviv, and the right side was done in, it's also left and right. No, it's the other way around. No, it's other way around. It was done in Bratislava. And you see that if you are antagonist of the FGF by receptor 1, you have dose-dependent decrease in alcohol intake, and also exactly the same shape dose-dependent decrease in dopamine neuronal firing activity, which really, I think, really show us that it's not coincidence. That there is very likely that FGFR1 mediated modulation of dopamine activity is indeed what is involved in alcohol intake. And we recently worked on 5-HT neurons, and it's not yet published. The study on dopamine neurons we already published about a year ago. And what's going on with 5-HT neurons is the opposite effect. We didn't see decrease in the firing activity, but we saw decrease in the burst firing of serotonergic neurons. And we still don't know it's also mediated via receptor 1 or not. We are working on it now. And finally, last topic before I will give floor to students who wants to present today, it will be about transforming growth factor beta 1. It's another growth factor. It's 25 kilodalton protein. Actually, it's not only, it can be defined not only as a growth factor, it can be defined also as a cytokine. It's released by leukocytes, and it controls proliferation and migration of immune, epithelic, and connective tissue cells. And in the brain, it's known to be involved in neurogenesis and axonogenesis. But we get interested in TGF-beta 1 because of the recent study, which found that it has antidepressant and anxiolytic effect in suspension test and forcing swim test and in sucrose preference. And what is very interesting, why I was get very interesting with the study, they use nasal administration of TGF-beta1. And we ask question again, if this antidepressant-like effect of TGF-beta1 is connected with monoamine systems of the brain. And we use the same protocol in the study. We administered it nasally under the brief anesthesia. We brief anesthesia of rats by isoflurane, and then we administered nasally TGF-beta. And then we try to check what's happened with dopamine and serotonin neurons, and we didn't found any effect. Which you may notice that in last years in many studies, I not so many times present noradrenergic neurons, because from our experience, we saw much less effect on noradrenergic. We did not see effect of, for example, of delta opioid ligands, of trace amines. Then we somehow neglected noradrenergic system. But when we did not see effect on dopamine and serotonin, actually my PhD student, Ruslan, he found articles that there is much more dense and short connection between locus coeruleus and olfactory bulbs. So maybe this is mediated by noradrenaline. And indeed, we found some decrease in statistically significant tendency to the decrease in the pharynx activity and statistically significant decrease in the burst activity of noradrenergic neurons. It's good news because now we know that nasal administration is working. It's supposed to arrive to the deep brain areas and to have effect. The less good news, we still don't know how to repeat it, because usually antidepressant-like effect should be connected with increase in monoaminergic activity. Maybe we have increase in extracellular noradrenaline, which suppresses noradrenergic neurons. And to check it, we need to go to check chronic administration of TGF-beta1, what we are probably going to do soon. And conclusion what we will talk today is that TLQP62, FGF20, and TGF-beta1, the antidepressant-like effect might be linked with monoaminergic neurotransmission. And I think I will finish by this. I can take your questions, and then we will invite who wants to speak today. You. Would like to start? Or some questions before?
Does it matter which areas, like the levels of all of these growth factors in the brain?
Yes, I think it's very important in which brain areas it is.
Yeah, but like there are some areas that.
It's very important in hippocampus, for example, in prefrontal cortex.
Yeah.
Yes.
Are there any adverse effects for this growth factor, as you said about the FGF2, about the alcoholism?
Yes, it's
for the TDE, is there any adverse effects or just
might be. I don't know, to be honest.
always another balance?
Yes.
And for now, in all growth factors, the better. I wonder what is the trade-off?
Well, it's a very good question. Yes, is all generally growth factors. We are still pretty far away from using of growth factors as therapies, and main problem is it's for example, you cannot give FGF to patients because it will be have CV. It's a very it's very not good idea to treat with the growth factors because it can induce tumor and
Yeah.
stuff like this. Definitely, we need its no growth factor itself will never become therapy. But receptors, ligands of the non-receptors, they might become therapy, but we also should be very careful. And together with, I think growth factors will become therapy when we will know how to deliver them to the very specific area of the brain. I think Hila is working on it. You're working on Matar. She's working, for example, Matar. She's work because FGF2 you cannot use for the treatment. I think she would like to use microRNA, which blocks its expression.
I'm very familiar with people with their thoughts in the lab, but I haven't.
Okay, please take the floor. You have disk. We have one participant in Zoom. Can I copy it and upload it to the course website? Don't be. Sometimes I don't know, but when I use Zoom, sometimes I have problem with no, okay. Presentation one. Can you type your last name, last name? BDNF, I'm almost sure that yes, but I don't, I cannot recall right now. I get to addiction really, and to start with addiction research very recently. In the Zoom, you see, do you see it?
No.
No. Wait, yes. Now?
Yes.
Okay.
I think this is this one, not this one, but never mind.
What's going on? Why we don't see it on our desk?
That's fine.
It's fine, but we don't see it on our desk. Your presentation. Wait.
We need to do that. So we can see what they see on the screen. Yeah, the slideshow. Oh, then they see that.
No. No.
Do you see the presentation?
No, we don't see it. Let's stop share. And we will share it again. I will close my better. Now. Let's go back, let's open it. We make full screen, we see it.
Okay.
No, but we don't see it here.
We'll see.
Yes, we see. Okay, and
Do you see it in the Zoom now?
do you see it?
I don't see it on Zoom now.
Okay.
I did
Let me
not do, well, I did not do sharing.
share this one.
This one.
Yeah.
Now,
Yeah, I can see it.
Okay.
okay.
So I'm going to talk about something that's related to my project and my business. Um, so in our lab, I mean, in Valenia's lab, our main research is sleep, but and we have like animal and human branches of the lab. In the animal, we focus on the noradrenaline system. And as I'm really interested in neurodegenerative disease, this is what I wanted to focus on, and I'm going to talk to you about it a little bit. So currently, the locus coeruleus is one of the most hurt areas, or it's really loses its volume in later stages of the disease. It gets to 70% of the cellular loss, which is a lot for like a normal elderly. It's something like 20%. So it is a big deal, and a very interesting thing that it's not like something that happens only in the end of the disease, but the reason for this cellular death is the tau accumulation. Wait, maybe I can do that. Okay, yeah, that's fine. Okay, tau accumulation. Apparently, this is the first area in the brain in which phosphorylated tau, which is one of the two major pathological biomarkers in Alzheimer's disease, are. And this phosphorylated tau is actually, you can see it in very early ages from 30 years old, which is much long before we see any clinical clue for that there is Alzheimer. And this is why it's a pretty good candidate for targeting the disease. Yeah, we can see here in Alzheimer patients. Well, it is a bit bluish. We can see here that here it's much larger than in the Alzheimer's patient brain, and there is more tau aggregations. And the fact that it starts to affect the locus coeruleus very early, but the cellular death is very in the end of the disease, makes it a very potent potential target for maybe treating or making the disease progress slower. And this is what first people saw in like postmortem evidence, and it's quite like new from the late 90s. Like the main focus of Alzheimer's disease was acetylcholine for a very long time, and the neuroadrenergic system is something that is a bit newer, and it's now with new technology we can see more in vivo, like in living humans, clues for that or evidences. So apparently the neuromelanin that is in both substantia nigra and the locus coeruleus is has specific magnetic whatever characteristics that helps it to be very prominent in specific MRIs, and this way we can see the alpha structure and how is it related to different symptoms of the Alzheimer's disease. And there is a correlation between the LC integrity and the cognitive performance. also between the LC integrity and nocturnal awakening, or let's like sleep quality in general. And also, this is like what I'm mainly going to focus on my thesis. And also, that was pretty surprising that there is a positive correlation, meaning the more the LC is in like healthy, or like this, the higher the signal, there are more neuropsychiatric symptoms, mainly talking about aggressions and which is might make sense, but it's not like less functioning, worse behavioral outcomes. And that's like, okay, we see that some structural changes are in these brains, but what's happening in the actual, like how does it affect the noradrenaline levels? So in CSF, only late stages we see an increase in the CSF, which might be a bit puzzling, like, okay, there is a disintegration of the area, so why we see an increase of the noradrenaline? And also there is increased expression of tyrosine hydroxylase, which is one of the enzymes that producing noradrenaline. And also, well, all these evidences are coming to toward an understanding that there is kind of a compensation mechanism, like that the high noradrenaline levels and the higher expression of tyrosine hydroxylase is to compensate for the cellular death of other LC cells. Like in the remaining cells, we see also that there are more axons ending and dendritic like spikes, how do you call it, spines, dendritic spines, and this is kind of a way of the body to cope with the loss of such a big part of the cells. What happens with the noradrenergic receptor is not really clear, but we'll talk about it a bit later. In animals, it's a bit more known or sure that at first we see this kind of compensation, higher rate of activity, and in the end, when there's no cell, of course, there is hypoactivity and lower levels of noradrenaline. So there is imbalance at first, like it's overcompensation, and in the end, it's like hypoactivity. There is not enough cells to cope. And it might be related because if you first overcompensate and you and the firing rate is higher, and you try to compensate, like there is hyperactivation, it actually causes further degradation of the cells. And why is it so important specifically? How does this system, okay, so it's a symptom, but how does it affect what you know about the disease? So apparently noradrenaline has a very big anti-inflammatory roles. It inhibits specific chemokines, it regulates microglia activity, and as we now think that Alzheimer's is really related to these neuroinflammation direction, it makes it much more clear that, okay, there might be a relationship to the noradrenaline. Also, it's really effect on BDNF production, and it's also bidirectional. So more BDNF, it's similar to what happens with what we learned with serotonin. We still don't know, but no, there are evidence that BDNF also increases leukocerulous cells activity. So once like there is less activity, less BDNF, less activity of the LC, like kind of a positive loop to the negative side. it also very much related, like I said, to the sleep and wakefulness regulation. And we also know that here there is a bidirectional connection. Like the more you sleep, the disease progression might be slower, because sleep is very important for the plaque cleaning, or all the like and generally regulating the brain states, maintaining cells. And once there is a change in the sleep patterns, it might actually make the Alzheimer's disease progress faster. And also one other thing is like neuromelanin that makes the cells like with color. It's something that it's actually a toxic particles that come from different heavy metals. I don't want to get into it, but the fact that these cells are containing it is very important for the body. As as a cell dies, it gets to all the area, and there is a much higher inflammation and immune activity. So it's not like it's a protective, but it's just containing it, and when it's not there, bad things happen. Yeah, in the substantia nigra and locus coeruleus. So now it became pretty clear. Okay, there are some. Let's work with that direction. And there are some clinically tested options. So the most primitive one, MAO inhibitors, didn't work so well. It was like just okay for the first few weeks. It seems like it actually makes the cognition better, but it then goes down. So they didn't proceed with that. It's like from the 80s. NRIs, they did pretty short, like it's 6 months. So they say we don't expect to see any behavioral outcomes, but we do see like biomarkers, positive signs. I don't know if like they kept doing it. I didn't find any research. And one more promising option is vagal nerve stimulus, which is this device. You put an electrode on the vagal nerve, and it's not really clear what it does to all the to the brainstem nuclei, but it is in animal we see that it increases LC activity and increases BDNF synthesis. And this one is very promising. There are like 3 or 4 clinical trials on that. It seems to improve scores in cognitive tests, not in like mood and sleep. They check that too. It doesn't help for this stuff, even though like VNS is something that's sometimes used for no, it's actually not, never mind. So these are like the clinical, or VNS is the most promising one, but there are a lot of like things that are tested in animal models. I wanted to talk about them just because they're a bit different than the one we talked about here. So L-DOPS is the synthetic equivalent of L-DOPA in dopamine. Like this is a version that can get into the brain and be synthesized into noradrenaline. And when combining it with NRI, it means more noradrenaline in the synapse for a longer time. And the problem with animal models is that specifically because the pathology of locus coeruleus is related to the tau, there are no really good models for that. Like all the animal models are about amyloid beta. And if there are models about like tau mutation, it's not something that really happens in Alzheimer's disease. It's like a model for a different disease. So we don't really know how can we mimic the hits the boot. Yeah, the accumulation of tau and how it affects in animals. So everything I see in animals is like, okay, maybe, we don't know. There is a bit more amyloid beta effect on the LC. Maybe it's totally different than what we see in people. But it does make things better in two different mice. In this one, in the second one, there is no actually production of noradrenaline. And it helps with in cognitive function. Another way is try to overexpress again tyrosine hydroxylase, which is kind of weird. Like, this is what we saw that actually happens in Alzheimer's disease. So we try to mimic it as a therapeutic intervention, and it seems to increase BDNF, some like positive signs of reduced accumulation, but it did something pretty weird for their anxiety. Like, it's like when you read a review about it, it's like, yeah, it's make their activity more normal. But when you look, you see that they were more explorative and less anxious than wild type animals. So I know it's very confusing, all these kind of tests, and when you say, okay, their phenotype is not normal, but is it really similar to what happens in what we're trying to mimic? Like, if they're more exploratory, maybe it's not what we're looking when we're trying to compare it to Alzheimer's disease. Yeah, and also it's something that is also like in treatment for a sleep disorder and MDD, which might be related because it's common symptoms. And the last thing is about the receptor modulation. So something that is like in line of what we said is, okay, we want to increase noradrenaline, let's block alpha-2, which is inhibitory receptor, and it actually like makes things better. But on the other hand, beta receptors, actually, if we inhibit them, it makes things better. So on one hand, we want to increase as much as we can in all kind of ways the noradrenaline levels in the system, but on the other hand, we say, no, for specific types of receptors, for example, beta receptors, activity actually increases the production of amyloid beta. So it's a very, very complicated mechanism. I really don't know what to think about it. Like, even like the fact that all these interventions are about elevating noradrenaline, even that I'm not sure this is the right direction, because dysregulation doesn't really mean, okay, we should do that very big intervention, and it's a problem. Like we said about the growth factor, as long as we can't target a specific area or a specific thing, it's really hard. And that's it. That's what I wanted to talk to you about. And I had this game word in like this word play in my mind, and I wanted to share it. That's it. Yeah.
Good question.
Yeah, of course.
What about the neuromelanin? Is there any connection between the sleep wash cycle of the brain, the theory that we need to sleep washing out maybe, or anything of that sort?
So it contained really secured in the cells because there is no really good way to wash them out. Maybe some of these like precursors or some of the heavy metals can be washed out, and then maybe less neuromelanin or something like that. But the neuromelanin itself is, as it's a compound, stuck there forever until like the cell die, gets messy.
And secondly, is there any, if you know, it's not definitely related to this, is there any tests about Alzheimer's disease and melatonin? Considering we know that it's physically connected to sleep cycle.
Yeah, it is like the Alzheimer's sleep disorder is kind of
Monthly.
no, it's very like it's basic, like it's something that everyone had. It's not something so specific as I can say, oh, this is Alzheimer, but there is this regulation of the circadian, and maybe melatonin can help. I don't know, I didn't search into that, but might be also interesting. That's it.
I also have a question about the neuromelanin. I just went from like another class, but I'm like not entirely familiar with it. So what I understood is that it's like a byproduct of dopamine
Yeah,
trash.
yeah.
So like the specific study I'm talking about, they use neuromelanin to a neuromelanin MRI to try to find Parkinson's, like biomarkers for Parkinson's disease. So I was just wondering, what does neuromelanin have to do with the lupus coeruleus?
So lupus coeruleus also produce dopamine.
Right.
Lupus coeruleus also produce dopamine.
Yeah.
And it's a precursor of noradrenaline, yeah. So it's also there, and I'm pretty sure that like the trash of the noradrenaline also gets into the neuromelanin compound. So yeah, it's the same. The signal is a bit lower than in the substantia nigra. Like the Parkinson, the connection between Parkinson and neuromelanin MRI is really, really strong. And now, like in the last few years, it's also became pretty strong also in the lupus coeruleus. Partially because we have now the much like stronger magnet, the T7 7 Tesla MRIs, and now we can see the signal much better.
But you said that the more it detects, the more neuromelanin detected, the more pathology there was?
No, the opposite way. Because it's something that we accumulate through life. It's something that is inevitable. But once the cell dies, this is the only way that they can be like cleared out. So if we're talking about older age, less neuromelanin means less cells that containing neuromelanin.
Was there anything similar done in people with sensitive behavioral disorder?
So REM sleep, like RBD is something that is a symptom of Parkinson's disease.
Like something that happens all of the things you're talking?
No, it's I've understood, yeah, all of them. But Alzheimer's is not a synaptic. Yeah, it's a very common like in 90 this kind of a disorder is pretty much moving through REM sleep, which is something that is not happening usually. Your body is paralyzed to make sure you're not acting out your dreams. And in this disorder, what happens is you do active dreams, and it's something that in 95%, I think, of the cases, it will mean you'll have Parkinson's disease, but it's not something that we see in Alzheimer's. I'm not sure like to what it is related. Probably not to the lupus coeruleus itself.
But I know it's somehow related to norepinephrine signaling that follows the
So the subceruleus area, which is just the subceruleus is I think it's pretty sure it's a glutamatergic area. And this one is causing the paralysis, and I don't know if like it's pretty, like we know, oh, this is the area that is really get rid or destroyed in Parkinson. But in animals, this is the area when you lesion it, you see the cat acting out its dream and trying to catch mouse and stuff like that. But I'm pretty sure that noradrenaline like directly is not really related. It's not something that you see when you lesion the LC. That's it. It's really, really complex. Like everything in this course, it's like we have clues, but when we have opposite of it, yeah. Thank you for the course. It was really interesting and really.
Very, very good presentation.
Thank you for all the time. Took longer than I expected.
Well, that small closing remarks, I would like to thank to everybody. I enjoyed your participation, active participation. Thank you for your interest. It was very difficult year for everybody, and this is why I'm especially appreciated. And I hope that we will find ourselves in some kind of collaboration in the future. As I told my lab, I have always philosophy in my lab so to have the open doors. If you think about some collaboration, just want to come, you're very welcome. And we have a pretty good programs which to visit Slovakia for research purpose. For example, we have national stipend program. And if some I will send message to everybody with the links. And it's program for everybody. It's starting from undergrad student until full professors. You can come to Slovakia from the period from 1 month to 11 months. If you think to do some collaboration to part of your PhD or master in my lab, will be for example, if you think to record, if you would like to try to do some noradrenergic electrophysiology and to edit. Okay, thank you very much, and now I'm waiting for your works. It's still end of October, but who submitted yearly will. I already get one work. This I will check and will grade immediately after the end of the course.
Very much.
You're very welcome.
Thank you.